Well, everybody, this is Jason from STEM Nerds. Today's video is really important. I don't care if you're into science, not into science. Today, we are going to be talking about how we control microbial growth. This is not just in a science setting. It can be in your everyday life. It's a really important video. I think everybody should know how to do this. And if you haven't seen how microbes grow, I'm going to put that in the upper right-hand corner as well as in the description below because that is really important. It's harder to understand how to control microbial growth until you know how they're growing. And so let's go ahead and take a look at how we do this. So first things first, there are a number of different ways that you can control microbial growth. You can use physical methods, things like filtration and using heat. There are also chemical methods, and we're going to talk about both. But before we do, we need to discuss some special considerations. If there are certain things in an environment, it's going to make it more difficult to control microbial growth or get rid of microbes. So the first thing we need to discuss is some microbes have the ability to form spores, endospores. These microbes go into a dormant state where their metabolism pretty much goes to nothing, and then they have this protective coat that shields them from environmental stressors, things like UV radiation, desiccation, drying out, antibiotics, antiseptics, disinfectants. And basically what this does is it allows the microbes to survive extreme environments in which they cannot grow, and then they will go into a vegetative state, metabolically active state, once the environmental conditions return back to normal or optimal. And so the reason why these spores are so difficult is, think about it, if you're trying to use antiseptics, disinfectants, UV light to get rid of them, well, you're probably not going to do that. They're even resistant to boiling. And so these can be a little bit of a problem. Certain types of bacteria can also be problematic, one of which is the one that causes tuberculosis. And the reason why, this is an acid-fast microbe. Tuberculosis has a special waxy coating over the cell wall known as mycolic acid. And this mycolic acid can help protect them. Not only that, but what we've talked about in the past is that tuberculosis also grows very slowly, and that could make it resistant to things like antibiotics. But there are other microbes out there that could potentially cause issues, one of which is known as the pseudomonads. And the pseudomonads are another type of microbe that just can grow in some really extreme environments. Sometimes you can find them growing in soaps and disinfectants even. Even certain viruses can cause issues when it comes to trying to disinfect an area. There are two main types of viruses in terms of their external architecture. One are the enveloped viruses, and then there are the non-enveloped viruses. This is a picture of an enveloped virus. This is actually an easier one to deal with because that envelope is a lipid. It's a phospholipid bilayer similar to what we have in our cells. In fact, most of these viruses obtain this lipid from the host cell. Now, because it's made of lipids, it's really susceptible to disinfection. The disinfectants will often destroy that lipid envelope where the attachment proteins for the virus are usually contained. And so if you destroy that lipid envelope, you destroy the attachment proteins. And if the virus can't attach to anything, it can't cause an infection. Now, the non-enveloped viruses, these are a little bit more problematic because the attachment proteins the virus uses to recognize a host, those are attached to the protein capsid. There is no envelope. There is no phospholipid bilayer covering that capsid. So when you add the disinfectants, they struggle to denature the proteins that are holding that virus together. And so the main point is that there are infectious agents that make it a little bit more difficult to disinfect an area. And if we know that going in, if we suspect that there might be non-enveloped viruses, some type of mycobacterium, some type of microbe that is a spore former, all of those things are special considerations. But it's not just the microbes. Environmental conditions can also play a role as to whether or not disinfectants will actually work. And so things like, well, I know this is kind of gross, but a lot of organic matter. And so I think a lot of us intuitively know if we suspect that there is infectious agents in organic compounds, the first thing we're going to do is clean up the mess, so to speak. And so here we've got a dog had a sick stomach. And so hopefully we wouldn't just spray a whole bunch of disinfectant in this organic matter and expect it to do its job. Most of the disinfectant will be bound up in all the food and everything else that is in this pile of organic matter. And so you clean that up first. all the bulk contamination, and then you spray it down with a disinfectant, and that will then allow it to work a lot better because it's not interacting with all the other organic compounds in the spill. And so that would be the same if you have vomit, if you have blood, any type of bodily secretion. You want to get that cleaned up first. But you also want to consider other environmental parameters, things like temperature. If it's really, really hot in an area where you're planning to use a disinfectant, one of the main things, one of the most important things when it comes to using chemicals is something called contact time. The amount of time the chemical is going to be in contact with the potential disease-causing agents. The less that is, the less effective that disinfectant is going to be. And so if you've got really high temperatures, usually the disinfectant is going to evaporate a lot faster, especially alcohol-based disinfectants. If you have extreme pH, either very basic or very acidic, that can break down disinfectants before they have the time to do the job that they were meant to do. So be aware that even extreme humidity, extreme temperature, changes in the pH of your environment where you're using disinfectants, that can all have an impact on how useful that chemical is going to be. Now, when it comes to disinfecting instrumentation, especially in the medical field, there are three main categories. The first one are non-critical instruments. This stethoscope would be an example. A non-critical instrument is one that is going to maybe potentially touch the surface of the human body, but it's not designed to go into the human body. And because it's a non-critical item, usually disinfection will work. Sterilization is often not necessary, which brings us to another concept. These words that we've been using, disinfection, sterilization, they have a meaning, especially in the biomedical field and in microbiology. And so a lot of people would say, hey, you know what? I think this particular item looks clean. In microbiology and in the biomedical field, that doesn't have a whole lot of value because when something is clean, it just means that the structure, or if it's closed, is free of visible dirt. But we know that microbes are something you can't really see. And so just because something looks clean doesn't mean that it is free of microbes. And so we have to be really careful with that term. Now, when we say something has been disinfected, that is a lot more meaningful because that means steps have been taken to reduce the number of microbes to some level often considered safe. Now, that can be different depending on the environment, depending on the type of structure that we're talking about or the instrumentation, but disinfection is different than clean, and it has meaning in the sciences. And then we have sterilization. When something is sterile, it is free of all potential disease-causing organisms. That's your bacteria, your viruses. Now, for me personally, that should also include all the prion diseases, those protein diseases, although in certain instances that is not necessarily included in some biomedical fields. But point is, sterilization is the highest form, it's the safest form of reducing microbes because the microbes should be reduced to basically zero in a sterile environment. So the next level when it comes to instrumentation are semi-critical instruments, and semi-critical instruments might be like an endoscope. And what's happening here is you've got an instrument that is designed to go into the body in the sense that it comes into contact with mucous membranes, maybe in the respiratory system or the GU or the GI. And here, ideally, it would be great if the instrument could be sterilized, but practically speaking, that can be very, very difficult to do. And so we're looking at least very high level disinfection with the goal of trying to sterilize it. But like here with an endoscope, you've got cameras, you've got electronics. And so as we're going to see in terms of the types of things that we have at our disposal for disinfection and sterilization, sometimes that can be a tricky one. The next level are the critical instruments. These are instruments that are designed to go into the human body, so things like scalpels and needles. If you've got an instrument or a structure that is going to be going through a physical barrier, skin or mucous membranes, here these things must be sterile. So critical instruments must be sterilized. All right, so how do we do this? How do we reduce the number of microbes, both in everyday life and in the biomedical fields and in microbiology? Well, again, here we're going to start out focusing on the physical methods of control, one of which is temperature. It's one of the reasons why we cook our food, because by cooking food, we introduce high temperatures and many microbes cannot withstand those temperatures that we use for cooking, and so they're going to die, and dead microbes are not going to cause disease in the human body. And so we have this form of a physical method, that is heat, and cooking would be an example of using dry heat. Now, there are others as well. Open flames would be another way of sterilization. Now here what we're looking at is a microbiology tool. It's called an inoculating loop, and we use this to transfer microbes from one type of media to another. And we really need to ensure that that tool is sterile before we start transferring microbes. And so to do that, we use an open flame, because the best thing about an open flame for the instruments that can handle an open flame is there is no living things that can survive an open flame, fire. And so we basically essentially just incinerate any type of contamination on that loop and make it sterile so that we can transfer microbes without getting any cross-contamination. Obviously here we're going to have limitations when it comes to using dry heat. The thing that we're sterilizing has to be able to withstand those really high temperatures. And so usually they're made of metal. There's not really any electronics. There's no rubber components or plastic components that could be melted. And so we have to be careful when we're choosing a sterilization or disinfection method that the structure that we are intending to disinfect or sterilize can actually handle the method that we're employing. We also have what's known as using moist heat. And so in this particular case here, we've got pasteurization, which we use for a lot of food processing facilities. And with pasteurization, what we're doing is we're heating, let's say it's milk, right? So some of our dairy is pasteurized, and so, or some of our juices. And what we're doing is we're heating those food products. And by heating those food products, we're decreasing the number of microbes that are found in those products. Now, they're probably not going to be sterile. And if you don't believe me, take a sealed carton of milk and leave it in the refrigerator for a few months. And even though it was sealed and nobody touched it and it was pasteurized, it will start to spoil. So there will be some microbes often in a pasteurized juice or pasteurized milk, but the number of microbes will be reduced to some level considered safe. And there's always a trade-off when it comes to using heat, especially with food processing. The more heat you use, sometimes, well, the food's not going to taste as good. We start to ruin some of the nutrients in that food as well. Boiling is another example of using moist heat to reduce the number of microbes. Now here, boiling does have its limitations. We talked about spores earlier in the video. Spores can withstand boiling. In fact, we used to isolate spore-forming microbes in one of the labs that I taught at. The very first thing we would do is we would take a soil sample, put it in a nutrient broth tube, boil it, and guess what would happen? It would kill off all the microbes, the vegetative microbes. However, the spores would survive, and then we would take that and allow it to grow in an incubator. And guess what? Once it cooled down and those spores were there, now they had optimal conditions, the microbes would start to grow. However, boiling can be a great way to reduce the number of microbes, especially associated with food. But again, we talked about limitations. When you're boiling something, sometimes, especially if it's vegetables, some of the nutrients that are contained within those vegetables start to break down and may even be released into the surrounding water. Perhaps one of the best ways to use heat in sterilization processes is right here. It's called the autoclave. And the autoclave works by superheating with pressure and steam. It combines those things together. And so often with an autoclave, we are heating things to 121 degrees or around 120 degrees Celsius. We're using high pressure, 15 psi, and we're usually keeping those parameters somewhere around 15 to 20 minutes. And when we do that, it generally wipes out just about everything with the exception of those infectious proteins called prions. But viruses, all types of bacteria, endospores, all gone. This is a really important machine for us in microbiology. After we've inoculated microbes and we've allowed them to grow and they're highly concentrated on nutrient agar plates or in broth tubes, we can put those plates and broth tubes in an autoclave and be ensured that pretty much everything, all the microbes are going to be dead after a cycle in an autoclave. Super important in medicine and in dentistry. For those of you who are into tattoos and things like that, a lot of times those tattoo shops will have tabletop or piercings. Will have tabletop autoclaves to ensure that some of the equipment that is reusable can be sterilized. This is, of everything we've talked about so far, the best way to sterilize something. But again, we have to make sure that what we're putting in an autoclave can withstand that high heat, steam, moisture, and the pressure. So all of our electronic devices, that's going to pretty much not be a thing here. But if you've got what we're seeing here, a lot of medical instrumentation, works out really, really well. Now, besides using heat, there are other ways to disinfect and sterilize areas, one of which is using the electromagnetic spectrum. Now, let's talk about this real fast. On the left-hand side, we have very, very long wavelengths. And so we've got FM radio and microwaves. And so those things, at least the radio waves, they're not going to really destroy anything. Microwaves can only because it's going to increase temperature. And so that's why we cook food in a microwave. Then we've got the infrared, visible light most of the time. Visible light's not really damaging a whole lot, but it's those three on the right-hand side that I want to talk about. UV, X-rays, and gamma radiation. These three come in really, really handy when we are trying to sterilize an area. UV light is great because it causes something called thymine dimers. So if you remember when we were talking about DNA and RNA, we said, okay, listen, we have these A's, T's, C's, and G's. Well, those T's, those thymines, if they're found next to one another in the genetic code, they can covalently bond to one another, and that messes up the genetic code. And so UV light, there are UV light generators that hospitals will use. There's some downsides to UV light, and we'll talk about that in a second. X-rays, a lot more energy, so it's a little bit more dangerous for human beings, could be potentially cancer-causing, but again, more energy, really good sterilant. And then we've got the gamma rays. And so gamma rays, very, very high energy, very short wavelength, really good at sterilization. And so some of the microbiology stuff that we use, at the prepackaged nutrient auger plates and things like that, the plastic itself can be gamma irradiated to ensure sterility so that when we add the nutrients, we don't already have some cross-contamination there. This is a really good example of how we use UV light in the process of sterilization. And so this is a lab hood. This is where people will work in a lab and they will wear their gloves and their goggles and they'll look like something out of a CSI sort of TV show. That purple light that you see there is a UV light generator. Now, UV is not inherently purple. We don't see, if UV is out there, we're not seeing a purple color. The purple is there just to tell us, hey, this light is on because, well, we really don't want to be exposed to high intensity UV light, just like we don't want to be sitting out in the sun for hours and hours and hours on end, especially if we're not used to it. So the color that we're seeing there is just telling us, hey, this thing is on. But that UV is great because anything on the surfaces is probably going to be destroyed. But that's the key here. UV has some inherent downsides, and one of the downsides is it doesn't penetrate solid objects very well, and so it really has to, whatever we're exposing to UV light has to be on the surface. Even if you look in the far right, far left-hand corner, you're going to see some pipette tips in those plastic containers. The UV light will not penetrate the plastic on the top, even though the plastic is clear. So UV light, really, really great, causes those thymine dimers, not super expensive. Again, irradiation, it's not something we want to be exposed to. UV light, we don't want to be exposed to it for long periods of time. X-rays, gamma rays, we really want to limit basically to zero gamma rays and X-rays only when absolutely necessary in medical procedures. You know, another physical method that we utilize as human beings is filtration. And here we've got an example of a filter system. This is a water filtering system, and filters can be very, very good. And I tell my students all the time, if I were to pick, there's lots of different ways. If you were in, let's say, nature, and we ran out of drinking water and there was a natural waterway, well, most of us know if you just drink water out of a creek or a river, there's a really high chance you could get sick. And so there's things you can do to minimize that. There are, let's say, iodine and chlorine tablets that can be very, very good. You can boil the water, but we've already seen that the spore formers, well, they're still going to survive. And so while that's great, it's better than just drinking water straight out of nature. That's not ideal. But filtration. There are these life straws that have their filtration is so tiny, the pores are so tiny, it catches just about all disease-causing organisms. And so I tell my students, if you're going to be out on a hike, you're going to be gone all day, these life straws, these filters, work very, very well in terms of filtering out all potential disease-causing agents. And so filtration systems, depending on the pore size, can be really great. Obviously, the larger the pores of a filtration system, the less that they're going to filter out. The more pores they have, the more they filter out, but the faster they clog. And this is true with air filtration as well. And so we have choices in our homes. We can use filters that are really great at filtering out dust and dander. However, they're gonna clog much faster, and they're a lot more expensive compared to the filters that have larger pores. They're gonna let a little bit more through. They just don't clog as fast, and so you don't have to replace them as often. This is really important when it comes to the biomedical field, especially in research as well. They will use filters that have very tiny pores that are going to trap bacteria and trap viruses and airborne contaminants. All right, so those are some of the more popular ways to use physical methods. We have heat, both dry heat, moist heat, and irradiation and filtration are all really important. However, there are other things that you can do, and especially around the house, using chemicals. Now, there are two main types of chemicals. There are antiseptics, and there are disinfectants. Both of them are involved in the process of disinfection, which we talked about earlier. Antiseptics can go on or associate with the human body. That would be things like mouthwash and hand sanitizer would be two really good examples. Disinfectants are designed to be applied to inanimate surfaces, desktops, countertops, lab workbenches, that kind of thing. And they're really not designed for us to be spraying them or being exposed to them on the human skin. Generally speaking, then, disinfectants are going to be harsher. They're going to destroy life a little bit more effectively than antiseptics will. But as a rule of thumb, and what I always tell my students, just keep in mind, the more effective something is at killing microbes, usually the more dangerous it's going to be for human beings. All right, so let's talk about some of these chemicals that are often used to control microbial growth. First one, I think one of the most common are the alcohols. Most of us are familiar with them. A lot of us have things like isopropyl alcohol, rubbing alcohol in our homes, and there are important advantages and disadvantages to each of the chemical methods we're going to talk about. And so I'm just going to kind of show you a few, talk about a few, and their general advantages, disadvantages. And so with the alcohols, advantages. Readily available, you can go to pretty much any health grocery store, or a Walgreens or a CVS, and you can usually find some type of an isopropyl alcohol. It's not very expensive, which is great. And for the most part, at least with the ethanol-based alcohols, they're not overly harsh in terms of the human body. In fact, again, we can find those in hand sanitizers. That burning sensation for some of the mouth rinses, well, that's an alcohol-based mouth rinse as well. But there are some things to consider. All right, there are some potential downsides. One, alcohols tend to have they struggle with contact time. The amount of time that the chemical is going to stay in contact with the microbes on a surface. And the reason why that's a struggle is alcohols tend to evaporate pretty quickly, even at normal room temperatures. And this is one where if you've got high temperatures, if the room was in the upper 80s or into the 90s, you're going to find that the alcohols evaporate very rapidly. That's an issue. Also, alcohols are usually going to be flammable. And so we have to be very careful about how we store them and where we use them. Third thing to consider is not all alcohols are the same. Again, the ethanol-based alcohols that we're using in the hand sanitizers, okay, those aren't so bad, right? We can use those actually as an antiseptic, but there are some that are going to be a little bit more toxic, and you really don't want those on your skin. And so you have to be careful about the choices that we make when it comes to alcohol-based antiseptics or disinfectants. Is a group of chemicals called the aldehydes. And you might be familiar with one of the most common, formaldehyde maybe. There's another one called formalin. And these are interesting because, remember what I said, I want to repeat it again. The better the job that chemical or process does at inhibiting or killing microbial life, potentially the more dangerous it's going to be to humans. And so I want you to think about something for a minute. You could take, there's still schools out there that have specimens that were preserved in formaldehyde from the 1950s and 60s. This is a long time ago, and yet those specimens still have not decayed. In other words, microbes have not destroyed those specimens. What does that tell you about this chemical? It tells you that there's a potential for formaldehyde in particular to be very toxic, and in fact it is. And for the most part, as we fast forward into the 2020s, when we're thinking about how we preserve specimens, a lot of times formaldehyde is not used anymore. It's a formalin-based product that has a low level of formalin as well as other types of preservatives that's going to be a little bit less toxic to us human beings. And so again, this is a great chemical if you want to destroy microbes, but we do have to be careful in terms of its toxicity. The next one is the biguanides, and this one here is often used as an antiseptic. You can see the chlorhexidine here, and so you're going to find these in things like soaps and possibly some type of mouthwash, that kind of thing. And so this is just a group of chemicals that is typically used as an antiseptic. And so when we're looking at safety relative to the others, this is one of the safer of the chemicals. Next up is something called ethylene oxide, and this is a gas. It's the first gas that we've talked about, and the gases are very important because with the gases, they can get into areas that a liquid typically would struggle to come into contact with. And so ethylene oxide can be pumped into chambers, and it can destroy microbes where the chemicals wouldn't really be able to reach. Ethylene oxide gas is a very dangerous thing. It's a great sterilant, but it is not something that you would want to have contact with as a human being. Your eyes, your skin, your mucous membranes. So this is a dangerous chemical. That's great if you've got an enclosed system and you can pump that system full of ethylene oxide gas. Destroys microbes where chemicals can't reach, but we definitely don't want to be in contact with this stuff. The other nice thing about the ethylene oxide is because we're not dealing with super high temperatures or a lot of water, well, now we can potentially sterilize instrumentation that has maybe some electronics, or it is going to be sensitive to high temperature, high pressure, or heat. So the halogens are an interesting group. The halogens are a group of chemicals on the far right-hand side of the periodic table, and they're just missing one electron. They just want that one extra electron to feel fulfilled in their life. And there's a couple different types that we're going to talk about. One of them is iodine, and iodine has been used for many years, decades, probably hundreds of years, as an antiseptic. And so when I was a kid, if you fell and you got a scratch, you'd walk around, oh, look, I got a scratch on my finger. And then you'd have a little brown finger because mom would go over there and put iodine all over it. And it would be used as an antiseptic to control microbial growth and prevent infection. There are iodine tablets that you can put in water to help make it safer to drink. Doesn't taste very good, but it will do the job. On the other hand, you've got a completely different type of halogen. And that's this. This is chlorine. Now, interestingly enough, we can take chlorine and we can put it in powders and tablets, and that can be a great disinfectant, especially when it comes to water. Most municipal water is disinfected using some type of chlorine or chloramine, and that's what makes it safe to drink. We put it in pools and at higher concentrations to help destroy microbes there, so that swimming in a swimming pool is safe. However, there's another form of chlorine in its pure form, and that can be really dangerous. And that's this, chlorine gas. This is the second gas that we're talking about. The first one was ethylene oxide, and chlorine is the next one. Chlorine is extraordinarily toxic. If you walked into a room filled with chlorine gas, you would hit the ground before you ever knew it happened, and that would be the end of you. So chlorine, incredibly good sterilant. Again, potentially used in areas where you can't have a whole bunch of moisture or really high temperatures. It can be corrosive, so we have to be careful there. This is a really high level sterilant, but you also have to be really careful because it's very toxic. And then we've got the metals, and this is really important because metals can interfere with microbial growth. And so you might find these in ointments that, especially for people who are third-degree burn victims, you're going to find this stuff in some types of deodorants as well, inhibiting microbial growth. Again, there's some toxicity concerns there, so it's not necessarily something you want to be exposed to in high concentrations for long periods of time, but it can be a very valuable method for reducing microbial growth. Ozone is the third type of gas that we're going to talk about here today. And just like we've mentioned already, because it's a gas, it can get into areas where liquids cannot. Ozone can be used as a disinfectant for water. And so sometimes water is ozone disinfected instead of or in place of high concentrations of chlorine and chloramines. And it's basically an unstable form of oxygen that's going to break down microbes and bind to them and cause all kinds of issues with their macromolecules. Then we have the peroxygens. The most important one is hydrogen peroxide. This, of all the chemicals we're going to talk about, is probably the most misunderstood. Why? Hydrogen peroxide is actually a better disinfectant than it is an antiseptic. And we mostly use it as an antiseptic. And let me explain what I mean. If you've ever gotten a cut or an ouchie and you put some hydrogen peroxide on that cut and you see all the bubbles, you're like, oh, look at the bubbles, it's working. No, the bubbles means it's breaking down, it's actually not working. And so with hydrogen peroxide, H2O2, it breaks down into water and oxygen gas. The bubbles that you're seeing are the breakdown of the hydrogen peroxide, and that's the gas that's gassing off. Now, when it comes to hydrogen peroxide, I'll give you a really good example. My son one time got bit by a dog, and he had a pretty decent cut on the end of his finger. One medical professional said, do not use hydrogen peroxide at all because it is a better disinfectant than an antiseptic. But the other one said, hey, listen, as long as you are soaking that cut in the hydrogen peroxide, it's going to work a lot better. And so my son would soak it, and it did seem to work a lot better. And certainly there was no infection there. And I'm sure that was one of the things that contributed to there not being an infection in what was a pretty sizable cut on his finger. However, hydrogen peroxide is used, especially in the food industry, because it's relatively non-toxic. Again, it breaks down into water and oxygen gas, and so it can be used for countertops. It can be used for instrumentation. When you don't want to have something that's going to leave a residue that's going to be dangerous later on, hydrogen peroxide might be the answer there. Then finally, you've got the phenol-based compounds. These might be found in some soaps and in some toothpaste. Depending on the concentration, this stuff can be a little bit rough. Sometimes they use the phenolic-based compounds as a preservative. Now, one of the issues I can remember when I was teaching microbiology at a school many years ago, the anatomy and physiology department had a lot of leftover phenol, and they wanted us to use the phenol as a disinfectant for our lab benches. And so after about a couple weeks of doing that, I said, time out, we're not using this stuff anymore because I was losing my taste. I was losing my sense of smell, not because I was necessarily being exposed to it. I wasn't drinking the stuff. Students were spraying it, but it was in a fairly high concentration. It really wasn't something we should have been using. And so we put an end to that very quickly. So with the phenols in low concentrations, yeah, you're going to find them in soaps and things associated with antiseptics. In higher concentrations, they are going to be a preservative, used usually as a disinfectant. And finally, if we're talking about how to control microbial growth, what about the preservation side? Because that is something that we deal with every day. And so we do things that maybe we don't put a lot of thought into, but we put things in the refrigerator and in the freezer, and so we control their growth that way. We're not necessarily killing anything by putting it in the refrigerator, but we're taking the microbes out of that comfort zone, their optimal temperatures, slowing down their growth considerably and allowing food products to last a lot longer. Now, the freezer, we're taking away liquid water, and that is huge. A lot of what we do, especially when it comes to food preservation, is removing liquid water from the environment because all living things need water availability. When you put something in a freezer, all the water is frozen. Nothing can use frozen water as a water source. The other thing that can happen when you put stuff in a freezer, I'll give you an example, sushi, raw fish. If they're not going to cook the raw fish, a lot of times what they'll do is they will freeze it. And what the freezing does is it causes water to expand inside of cells. And if there are microbes or parasites that have been associated with that raw fish, once it freezes and that water expands, it can lyse those cells, thereby killing the infectious agents. And so that's one way to keep a product like sushi, if you're not going to cook it, a little bit safer. Now, that can happen in our home as well. When we freeze stuff, that in and of itself can help reduce the number of potential viable contaminant agents once the food thaws. Obviously, we've already talked about cooking food, raising the temperature in the other direction, right? And that will help destroy microbes. There's other things like, I always ask my students, a lot of kids will take a jar of peanut butter and they will just eat right out of that jar, and then they put the jar right back in the cabinet. That's gross. I get it. They're not supposed to be doing that, but people do it. And yet there's not a ton of microbes growing in that jar of peanut butter sitting out at room temperature that's clearly been exposed to people's fingers and they're eating right out of there and whatever. Well, why? Why do the microbes not grow? Again, it's available water. There really isn't any in a jar of peanut butter. Whatever moisture is in there is a combination of lipids. There's tons of sugar. And what we basically have is an extremely hypertonic solution that microbes really can't survive in. It's the same reason why we dry foods. You have things like beef jerky and that kind of thing. Using a lot of salt. Using a lot of salt will create a very hypertonic environment that, again, reduces water availability. And of course, there are certain chemical additives that can be put inside a food, things like nitrites and nitrates that make it harder for microbes to grow. All right, everyone, so that was a brief overview of how we can control microbial growth using all kinds of different methods. As always, thank you so much for being here. We'll see you next week.